Yeah, so in the end, we'll discuss the questions. So, are those questions related to the problems or some other questions related to the exam and all? Okay, so we'll keep it to the end, then we'll discuss about it. Okay, so and there will be also another session on Thursday in which we'll be revising all those topics. So if you still feel any doubt in any of the week sessions, then you can ask on Thursday as well. So let's start with the session. So this is your last week session, and in this week 12, we've looked into the ethical concerns about the germline gene editing, in which you looked into the bioethics, biosafety, and different regulatory issues in genome editing, and what is the need of those regulations and rules. And then we'll practice MCQs, and we'll also look into the current literature. So, first of all, we'll talk about how this bioethics originated, how the concept came in. So, it was coined by Fritz Zehrer in 1926, who was the German pasher and philosopher. So, he published an article named Bioethics, a review of ethical relationships of humans to animals and plants in 1927, and he introduced the concept of the bioethical imperative, which is the moral responsibility to all forms of life. So previously, as you have seen that uh, when we were during our bachelor's and old times, so people used to work on the animal models, and they used to kill those animals inhumanely to just study. But later on, it was realized that they are also life forms and we cannot exploit them. So later on, rules came up, regulations came up that you cannot exploit any form of living organism. So you need to keep certain things in your mind, that what are you experimenting on them. So later on, he extended the Kant's categorical imperative beyond the humans. So not just human subjects, but other animals and other life forms are also important. So we need to keep ethical things with respect to them as well. So according to Kant, because he was extending the Kant's categorical imperative, so according to Kant, he mentioned that do only what you believe would be right for everyone to do. So in other words, he said, so this was his quotation in which he said that there is therefore only one categorical imperative, and it is act only according to that maxim by which you can at the same time will that it should become a universal law. So it's complicated to understand from these lines, but what it says is that treat the people as valuable in themselves and not as tools to achieve your own goals. And his ethics focus mainly on that how the human should treat other humans. So these things means talk about the human subjects and keeping means human as a valuable entity. But why it started, because during the world war people were exploiting humans only. So at that time they are not talking about animals as of now, because that time humans were getting exploited for the experimental purposes. So that time this ethics came up for the humans to protect them. So there was another person who was Van Rensselaer Potters. So these people also he also contributed. So in 1971 he wrote a book named Bioethics: A Bridge to the Future, and then he also proposed global and interdisciplinary ethics, integrating both the bio, medicine, and philosophy. And he viewed the bioethics as a bridge between science and human values. So means whenever we are doing experiments we are scientists, but at the same time we should not forget the moral values and other things. So he said that bioethics are like a bridge between both of them. We need to balance both of them, and we cannot exploit any human or any living form by on the name of science. So he promoted humility, responsibility, and global cooperation in science. So one such example which I can give as of now is that for now many patients are suffering from many disease. So if I want to study a disease and if I am a researcher, so I need that data. I go to hospital, collect that data, and work on that, and means release some paper. But at the same time I cannot means make that data public. I cannot make the information of those patients public, because their information can be exploited by other organizations such as insurance companies or other companies who can threaten them. So as of now, So this thing that I can share the publication but I cannot share somebody's private data, so that thing is regulated by the bioethics. So means that is how the bridge, it acts as a bridge, that it is helping us to study something but at the same time we are also protecting somebody's right. So right to privacy is one such right. So these are his books, and then later on many people who means simplified these books into normal reviews or papers. So according to ancient Indian medical ethics, so means ethics is not very new term. In previous and means scriptures and other literature also these ethics were discussed. So Charak and Shushruta Samhita, which were written in 600 BC. So these were the earliest codes of medical ethics. So in them they also talks about the medical ethics. They emphasized on the truth, compassion, confidentiality, competence and service, and physicians viewed as moral guide, not just healers. So these things are also mentioned in those Charak and Shushruta Samhita, and till now it is relevant because it says that we should take the consent from the patient, end of life decisions and organ donation. So in all these things we still require the consent of them, and we cannot make that data public. So there is some regulations which exist in all these things. So if we talk about Shushrut, so he's Shushrut and he's Charak. So first of all, who was Charak? So he was father of Ayurveda and he has written Charak Samhita. So on the means name of this particular scientist, the book he wrote was Charak Samhita, and in which he explained about the digestion, immunity, mind body balance, and he focused on the Ayurvedic science. Then there was Shushrut, and he was also means near 600 BC only. He is known as the father of surgery, and the book which he wrote about the surgical procedures, how we can do, what kind of instruments can be used, so that is known as Shushrut Samhita. So based on their names only, their books were named. So in those books also it is mentioned that we need to focus these things, and we need to means the person who is a physician who is dealing with those patients, he should follow the moral rules and he cannot exploit the patients or people. So means this ethics term is not modern, it was also mentioned in primitive literature. And then in 1947 the study which means made people realize that how important these regulations are was the Nuremberg trials and code. So according to Nuremberg code, it says that means these code came after the trials. So followed the Nuremberg trials. So trials is like where the people are given punishment on the basis of the crime they commit. So this code was introduced in August 1947. So Nazi doctors were convicted of war crimes involving the human experiments on concentration camp prisoners. So if you have looked into the means if you have read the history and all those things, then you might realize that during the time of Hitler, what they used to do, those Nazis, they used to means ill treat the Jewish people. They used to perform experiments on them in the concentration camps. So that time they exploited many people during the World War without taking any consent from them. And even those people didn't realize that many experiments were performed on them. So after the World War ended, these trials were conducted in which many doctors were arrested, they were given punishments. So that time the Nuremberg Code came up in which they mentioned that what rights should be given to the people. So it consists of total 10 points. In the next slide we'll discuss all those points, but on overview it talks about the informed consent, benefit must overweigh the risk, participants understanding of possible risk. So if you're performing some experiment on some people by taking their consent, but at the same time you should also tell them that what are the risks which are associated with those experiments, and then how the experiments must be run. So this information should also be given to them. So the code was an attempt at establishing the clear rules about what was legal and what wasn't when carrying out the human experiments. Okay, so now we'll talk about that what was the Nuremberg trials. So it was the trial of Nazi criminals at the end of World War II. So the criminals, we mean the people who were performing the experiments on the human subjects without taking any permissions. So it was done in two separate trials. In the first trial, the aim was at the major crimes, and the second trial was about the lesser crimes which were committed. So four major charges were there when they were doing these trials. First was conspiracy to wage war. Second was waging in the aggressive wag, and third was the war crimes, and fourth was war against humanity. So this was one of the picture which was taken during that Nuremberg trials. Okay, so these are the 10 points which came up after the Nuremberg trials, because means when the crimes were seen that these are the crimes which were performed. So after that different code came up. So there were total 10 code. So first was voluntary informed consent, that the person on which we need to perform the experiment, that person should voluntarily give consent. We cannot forcefully ask him to give the consent. Second is fruitful result for the good of society. So if I am performing some experiment, then the result should be fruitful. It should not be like useless experiment that would harm the society. So it should be having the fruitful result. Third point was prior experimentation on animals and prior knowledge of the problems. So I cannot directly perform experiment on human subject. Before that, I need to perform it on some model organism or some organism who is more close to human. And then I should also have the knowledge of all the problems or side effects which those experiments can have on the human. Fourth is avoidance of unnecessary physical or mental injury. So for example, if I want to see what is the tolerance power of a human to electric current, so in that case I need to give current to a individual. So this is just an example. So I cannot perform such experiments which will give pain or mental injury to a person. So that is very wrong. So this is also that we should not give unnecessary physical and mental injury to the human. Fifth thing is banning of known lethal or disable procedures. So lethal means that can lead to the death of a person. So those procedures are banned or either disabled. Okay, so six is degree of risk should exceed the benefits. So if there is risk associated with that experiment, so that experiment can only be performed if the benefit is very high. For example, I'm testing my drug on the cancer patient, and that particular cancer patient in its final stage of disease, and we know that he'll die. So I can perform experiment on him for the testing of that drug if I know that my drug is promising, that it will lead to the cure of that particular cancer. So at that time I can perform experiment on the person. But if I am not sure about the drug, that whether it will work or not, and I am performing the experiment on the person who is just in the first stage or middle stage, so that is very wrong. So means there is high degree of risk. So in that case we also need to keep in mind that what is the degree of risk versus what is the benefit, and what is the probability of that benefit. Seventh point is proper preparation and proper facilities to prevent the injury or death. So during the experimentation when it is carried out, so we need to make sure that it should be carried out in such an environment that it should not lead to the injury of that person or death of that person. Eighth thing is performance of experiments only by the scientifically qualified persons. For example, if I want to means transplant some organ, so I cannot means some organ which is developed in lab, so I cannot allow any person who is not even qualified enough to do that surgery or do that experiment. So those persons are prohibited. Only those people are allowed who are scientifically qualified for performing those experiments. Ninth point is participants may freely end the experimentation. So at whatever stage, if participants feel that they are not uh, feeling good about that particular experiment, then they are free to end that experiment at any point of time. 10th point is the experiments must stop if it proves too dangerous. So at any point of time, if we think that our experiment is at that stage of time when it has got into very dangerous stage, when person might die if I continue that experiment, so we must have to stop that experiment. So these are 10 points of the Nuremberg Code. And then different conferences also were conducted later on. So in 1975, the Esilomer conference was conducted. So it convened to address the risk of recombinant DNA research. So first thing was about the ethics which we need to keep in our mind before performing the experiments. Now in this conference they are talking about how risky is recombinant DNA research. So scientists proposed self-regulation instead of government bans. So even if government is not putting ban on certain experiments, but still at the same time scientists should regulate certain, means self-regulate those things. If a scientist know that this particular experiment could be harmful, for example if I am working with some virus which if get means get out of the lab, then it can prove havoc. So at that time I should be very means what we say, I should be mindful of performing that experiment, even if government has not put ban, but still I should regulate it, I should not perform it if it is very risky. So that is this point, and it established the biosafety containment levels and guidelines. So this biosafety containment is very important, because when I'm working with some bacteria, let's say E. coli, then I can work even without any containment area in normal lab conditions. But if I'm working with very harmful or lethal virus or something very dangerous organism, so in that case we need to ensure proper biosafety conditions. So there are many guidelines which exist. We will not go into their details. So those biosafety guidelines exist and we need to follow them. So it marked the beginning of biosafety as a discipline. So after this conference, all these points were discussed, that what are the important things to keep in mind during the recombinant DNA research, because many times, so till now also in different sessions we have seen that how the ZFNs, talents, CRISPR-Cas9, all those things help in gene editing. So to transfer them into the means cell or body, we need some vectors. So in vectors we also use viruses. So at the same point, if we do not use them properly, then they can have side effects and means adverse effects also. So at that particular point of time, these biosafety rules are very important. And this is a means essay in which the meeting points were discussed. So this meeting was conducted by Paul Berg. So it is known as Acylomer 1975, DNA modification secured. So if you're interested, you can read this article in which they have talked about that what were discussed during this meeting. So it was the California meeting which set the standards, allowing the geneticists to push the research to its limits without endangering the public health. So means we should perform experiments, but not at the cost of health of the normal public. Organizer Paul Berg asked if another such meeting could resolve today's controversies. So in later slides we'll also see that means what were the different tragic incidents which took place. Means although they were not documented, but still they were discussed. So before that, we'll talk about the scope of bioethics, that what comes under this. So it encompasses four main areas. First is medical ethics, second is research ethics, third is environmental ethics, and fourth is public health ethics. So in medical ethics, it talks about the patient rights and clinical decisions. In research ethics, it talks about informed consent and safety. So research ethics means if we are experimenting, we are performing some research on the group of people, maybe to study their psychology, behavior. So in that case, the ethics which are followed are research ethics, in which we need to take the informed consent, and we should also discuss about the safety with those people or the group of people on which we are performing experiments. Then comes environmental ethics, in which we need to keep in mind the human and nature balance. If I am performing experiment on the environmental things, then this the ethics which talk about it is environmental ethics, and then there are public health ethics which talks about equity, social responsibility. Okay, then there are many other ethics which lies in other fields also. Ethics is not specific to just the biology, it also extends to business ethics, environmental ethics, social ethics, organizational ethics, IT ethics, and many other ethics which exist. So in bioethics, different other ethics also means exist. This is not just limited to these four ethics, but we are mainly talking about these ethics because this is the scope of our session. But there are other ethics also, just like nursing ethics, resource allocation ethics. For example, if we have different resources, we have different therapies, so who can afford it, or means to who can access it actually, so that comes under resource allocation ethics, that the resources should not be limited to few group of people, it should be available to means wide group of people. So means the ethics which is related to that comes under resource allocation ethics. And resource means, I'm just giving example, resource can include different things such as vaccines, treatments, or health facilities, or many more things. Okay, then there is another means in one article this was discussed and I felt like it's important. So if there is a research question and we know the implementation problems, then different things could exist. So context and resources. So in that case, firstly we'll talk about the ethics. So in ethics, the things which come in related to the research question is the study design. Then, what is the choice of control, protection of participants, use of data or findings, and how to scale this up. Second thing is, who are the stakeholders? So, we are discussing different components which are associated with the research. So, first was the context and resources, in which we discussed about the ethics, which means before performing some research, these ethics should be kept in mind and how it should be implemented. That is second thing. And third thing is that who are the stakeholders who will be suffering from all these things. So policymakers are the stakeholders, means who are having some hold to that particular research problem. So first is the policymakers, second thing is the researchers, third are the program implementers, and fourth are the study participants. So these all are the stakeholders in this particular research. And what will be the outcome? So effectiveness, acceptability, adoption, appropriateness, visibility, fidelity, cost, penetration, sustainability. And then out means after means discussing all these components, then comes the dissemination, use, and iterative refinement and scale up. Okay, so these are the few points such as pragmatic trials, effectiveness implementation, hybrid trials, quality improvement studies, participatory action research. These all are the things which are done after means all these components. So this talks about that whenever, for example, I want to perform some research, I have the research problem with me, and I will be having the research question which I am going to ask. So what are the different things which we will keep in mind? Firstly, what I will ask, that what is the study design, what is the control, and who are the participants, what kind of data I am going to use. Scale up means if I am going to perform the experiments at the small level and I want to extend it, so that means I am scaling up the experiment. For example, to test the effect of the drug. So if that drug works, then I can increase the production of that particular drug, so that comes under scale up. And this is one example, that could be any other example as well. So I will keep these things in mind. Then I will also look at the stakeholders, means that will also be considered, that who are the people involved. And after that, we will see that we will perform some trials, we'll see whether it is effective or not effective, we'll check its quality, and we will also see the how the people are participating and how they are working towards that particular area. So after that, different outcomes could be achieved, which could be effective, or all these should be the outcomes. And then I can go for the further refinement and scale up after this everything works well. So these are the things which are kept in mind during the experiment. Means I cannot just means go and start the experiment before looking into all these means areas. Okay, so now there are different ethical concerns in genome editing. So means why they are concerns, because that in that particular conference also when we talked, so what are the ethical concerns in genome editing? So first thing is germline editing, because it alters the DNA which is passed on to future generations. So whenever I am editing some cell, so if it is not the somatic cell, if it is a germ cell, then germ cells are the ones which get inherited, which get passed on to future progenies. So if I am editing the germ cell, then there is chances that that particular DNA will be passed on to future generations, and you never know how it work out in future generations. So it raises moral issues about human identity and natural order. So for example, if I am doing the experiment in such a way to, for example, if a lady comes and she says that I want my child to be tall or to be fair or to be intelligent or smart, so there are different studies which talks about the genes which are linked to all these traits. So if somebody performs the experiment in such a way, they are edit the germline cells that the future progeny is of that sort, so in that case the human identity, which is the normal human identity, is lost, because we are customizing that human, and we are also disturbing the natural order. So these things raise the moral issues, because these things are not allowed. We can perform germline editing if it is very important, if the child, if we have identified that the means the parents were suffering from some disorder or some disease which is which is the X-linked, which shows the X-linked inheritance, or some germline inheritance. So in that case, we can edit it so that the child do not suffer from that same disorder or disease. So that thing can be done, but those things which interfere with the identity and natural order, they should be prohibited. CRISPR made editing easier, renewed the ethical scrutiny. Scientists called for ongoing global dialogue. So these are some of the points. Okay, so there is a difference between the somatic cell editing and germline cell editing. So this is just a difference, that how from the patient the cells are collected. So this is the adult patient from where the cells were collected, and then the healthy adult with edit in the liver only, and this is the healthy unedited adult. So if both are, means in this case if mother and father, so one parent is having the edited version and other is not having any edited version, but the children, they do not inherit the edits. So this is the somatic cell editing, in which the at the somatic cell level we have performed the editing. But in the case of germline cell editing, if we have performed at the level of germ cells such as egg or sperm cells by using the CRISPR Cas or any technique with which we have edited the that particular cell. So in that case, whenever the, for example in this case, the healthy adult is having the edits in all the cells, and this is the healthy unedited adult, so their children will inherit that particular germline edit. Yeah, so this is what they have discussed, that because previously on the germ cell when the child was about to born, so in that case at the germ cell level editing was performed, so he, means the unhealthy fertilized egg was converted to healthy fertilized egg after the germline editing, so that led to the growth of the baby who is healthy. But in that case, because germ cells were edited, so that entire baby is having the edits in all the cells, and then when he or she, whatever, marries the person who is having the unedited adult or who is unedited adult, so the progeny will inherit those changes, because in germ cell the changes lies. So that is the difference, although it got complicated, but it's just a one line, that in somatic cells the editing is not inheritable, whereas in germline cells the editing or the changes are also inheritable. So different ethics of gene editing are: gene editing power. First of all, it is very powerful. The technologies like CRISPR, they offer the ability to change the DNA and treat different genetic diseases, but there is a promise along with the concern. So while gene editing can transform the medicine, but still it also raises the concerns about unintended consequences. So for now, we don't know that what will be the side effect after 10 years. Even if we perform the clinical trials, but still they'll, means we can predict up to certain limit, but we don't know in future what would be the result. So in that case, we don't know about the unintended consequences. Although the beneficial results are seen, but still it comes with some consequences which might not be good. Then comes designer babies. So the possibility of using gene editing for enhancement, leading to ethical dilemmas around the designer babies. So this is the same concept, that when we are customizing those things and we are designing baby by ourselves, disturbing the natural genetic material, so that comes under designer babies. Equity and access. So it will only be, means affordable by the wealthy people, because who will be having the access to these life changing gene therapies, and deepening the societal inequality. So if these things were allowed, then what will happen is that, because these genome editing procedures, gene therapy, it is very costly, it is not easily affordable by the poor people. So what will happen is that the people who are rich, they want that their child should be best, they will go for the designer babies, they will perform genome editing, and all those things will be carried out on them. So it will lead to the imbalance in the society, because the people who will not be able to access it, they will be, means in the on the other side, and these health, means rich people will be on the other side, so it will lead to the societal imbalance. So in that way, to avoid that inequality, we should also have the ethics, so that it should, means these things should not be exploited by only one group of people. Then comes ecological impact. So how might altering the organisms impact the ecosystems and biodiversity? For example, we all know that there is a food chain, food web, all those things. So every organism depends on the other organism for their food and other things. So there is a balance in our environment. If I go and edit one thing, then it will also have its domino effect kind of a thing, in which one thing if affected, then it will also affect the other thing as well. So that comes under the ecological impact. So there was one story, I'll not talk about it, but for example, if I interfere with one breed of organism, then there is a possibility that that particular breed might also affect the other organisms which were dependent on that particular breed of organism. So in that case it will lead to the imbalance, and same goes for the plants as well. So what is our role? So if the means in this, the future biotech leaders who are working with these editing things, they must engage in the ethical discussions and responsible research and policy advocacy. So they will also, means they should also come up with the policies, that what should be the policies which must exist to perform the experiments without any, means troubles or harm to the society, and at the same time maintaining the balance as well. So there is a review about the ethical issues in the use of CRISPR-Cas9 in the creation of human disease models. So in this particular review article, so the conclusion was that one of the primary ethical issue is the potential for unintended consequences which could have serious long-term effects on the individuals and their offsprings. So although CRISPR-Cas9 means it is a talked about technology which is lot more talked about in terms of gene therapy and genome editing, but at the same time the main concern is that it could have unintended consequences. So in, in that sense it should only be used if it is very much needed, if its risks are low and benefits are very high. So that is what was discussed in this particular review, that why the ethical regulations are required in terms of the CRISPR-Cas9 with respect to the human disease models. Okay, then there was a graph which talks about the problems. So gene editing with the CRISPR-Cas9 creation of human disease models, it raises many ethical challenges such as safety concerns, then lack of acceptance, informed consent in which we need to perform, means inform the person on which we are performing this experiment, and potential risk of misuse, so anybody can misuse these things. Then fear of off target effects. So in this case, because CRISPR-Cas9 also, what it does is that it perform double strand breaks, but for that reason it need to identify the, it has the guide RNA which helps it to identify the region where it binds and where it need to perform the double strand break. But what if instead of binding to that particular site it binds to some other site? So there is a chance of the off target binding, and in that sense if it binds to some other region, then it might perform the double strand break on the gene which was functional. So in that case it could have the adverse effects. And then effect on the environment, so we don't know what effect it could have on the environment. For example, if I um, perform the genome editing on the organism to just increase the production of milk, for example I perform the experiment on the cattle to increase the milk production, or I perform gene editing of the, I edit the genome of the sheep or some cattle with the gene for growth factors or other, means other genes which could act like a supplement for the human if that particular milk is given. So in that sense it could have some effect on the environment as well. So these are the problems that are associated with the CRISPR-Cas9 gene editing. Now the methods, so well-defined regulations and ethical framework are required. There should be policies, there should be improved surveillance, because whenever the experiments are carried out we must have the proper surveillance that what is going wrong in what direction. Then maintaining the transparency, we should not hide anything. Same goes for the scientists, the policymakers and the patients, so no one should hide anything from anyone, it should be transparent. Then guidelines on the proper usage and potential harms, legal rules and regulations, and improve involvement of the stakeholders, so everyone should be involved in these things. So these are the methods which were discussed in this review, that how we can avoid those problems and what are the things which we need to keep in mind, and then what are the outcomes. So ethically bound usage of CRISPR-Cas9 technology, so if there are ethics which exist for the usage, then what can happen? So it provides the assurance of safety, because everything is now studied, that what could be the potential, this means harms or all those things. So it provides the assurance that okay, so this particular experiment is safe. Then increased acceptance, so because if it is safe then many people will accept it. Then consensual process, in which the diminished risk of misuse, because it is a consensual process, so misuse will also be limited. Then comes positive attitude towards the gene editing, strict legal actions on the exploiters. So if somebody is exploiting these techniques, then strict action will be performed on them. So in that way, all these things comes under the ethical practices related to CRISPR-Cas9, which can lead to the acceptance of this technique by many people, and which can reduce the ill effects or the harmful effects of this technique. So global regulation landscape, that how different countries looked into it. So 14 countries, they ban or restrict the germline genome editing, because as we discussed that these means cells, because they got in that get inherited, so these edits can also get inherited, which can affect our future progenies. So that's why these countries, they have decided that they will completely ban it. So these countries which are shown in the darker peach color, they are the countries which completely banned this. Then there are some countries which have some guidelines, but although they have banned it, but there are some guidelines which exist. So these are the light peach colors. And then there are countries which have some restrictions, that you cannot freely perform, but there are some restrictions, although you can perform those experiments. So these are the countries with some restrictions on the genome editing, which are shown in the light gray color. And these dark gray are the ambiguous. They are like maintaining the balance, perform genome editing, but with some guidelines that need to be followed. So major concern is the irreversible hereditary changes, because if the germline cell is edited, then you cannot reverse it back. So that is the irreversible hereditary change. So that's why this particular germline genome editing is banned in these countries. But that doesn't mean that all the genome editing is banned. It's just about the germline genome editing. The other genome editings are still allowed at to some level. So public opinion divided between the medical promise and the ethical risk. So because people are the stakeholders in it, because you are performing experiments on the people only, so there is the mixed opinion of these people based on the medical promise and what are the ethical risks. So based on that, some people support it, some people do not support it. Okay, so this is again the distribution among the 96 countries, that how they look into this particular germline genome editing, how many countries, which means so there are 56 about which the relevant information is not available, and remaining 40 which we have seen in this map. So 19 of them, they have prohibited it, 11 have permitted this, 6 are like indeterminate, and 4 are prohibited but with some exceptions. Okay, so what are the core ethical principles? So in NASEM 2017, these core ethical principles were given. So there are 7 core ethical principles. First is that the promote well-being, that is beneficence and non-maleficence. Second point is transparency in the scientific communications. Third is due care and deliberate application. Fourth is responsible science and global standards. Fifth is respect for the persons and autonomy. And sixth is the fairness and justice in excess. Seventh is international collaborations. So these are the 7 main points of the which are referred to as ethical principles. Okay, so with autonomy we mean that freedom to make the personal decisions and self-governing. The person on which the things have need to be performed, so they should be autonomous towards it. They are free to decide whether they want to be the part of that experiment or not. Beneficence means that that particular experiment, it should benefit the other people, it should be for the good. So in this particular screenshot which I have attached, so it is not just specific to the biological principles or the biological experiments, but it is just the ethical principles which are in general, means not limited to the biology but for the other fields also, because in the previous slides we have seen that ethics are not just limited to biology, it also exists in society, industries, companies. So means basic ethical principles are these. So what are the things? So non-maleficence means avoid doing harm, even harm that occurs while doing the good. Even if we are performing some good thing, but that doesn't mean that still we can cause some harm. So even means harm is coming at the cost of doing good, but still it should be avoided. Veracity means we need to tell the truth. Confidentiality means that we cannot share the private information of the people who are taking part in that particular thing, experiment, or any other thing. Justice means it should be fair, equitable, and appropriate treatment. So if injustice is done, then means it is not allowed. We cannot do injustice. It should be fair. Then fidelity means that we are faithful and practice of promise keeping and social contract. So means with those people who are taking part in the your experiment, so with them you should be very faithful, you should be loyal, and you should keep your promise. And then privacy means that particular individual has the right. So if that person wants that his data could be made public, so then only you can make it public. Otherwise, that person owns the rights. So that is, the data is private or it's very personal. Okay, so safety and risk concerns related to the gene editing. So off target effects which we have seen related to CRISPR Cas9. So it can lead to unintended mutations elsewhere in the genome. Then mosaicism is the uneven editing among the embryo cells. Germline editing not yet proven safe or necessary. IVF plus PGD remain safer alternatives. Okay, so there were some pros and cons related to this. So pros are like therapeutic potential. So these genome editing methods, they have the therapeutic potential in which we can correct certain mutations if they are, means those mutations if they lead to some disorder such as Alzheimer's disease, in this case example is given. So in that case what we can do, we can correct those mutations by using the genome editing tools such as CRISPR Cas9, TEL, and ZFN, although the CRISPR Cas9 is much more commonly used because it's better than others. So in that case it has the therapeutic potential. Isogenic cell lines, so it ensures that the differences are due to the specific mutations by eliminating the genetic variability. Drug screening, so it also facilitates testing the therapeutic compounds and identify the new treatment targets. Then personalized medicine, because with the help of genome editing we can do the personalized medicine in which we are taking the cells from the patient only and then edit them, and then again after editing we allow them to proliferate, and then we put it back into the same patient, so those T cells. So that comes under personalized medicine. That is the advantage of this. Then accurate modeling, similarly accurate modeling also, because we are taking the sample from the same person, same patient, and then we are treating it, so it's very accurate. But the cons are, the problems which are associated is the clinical challenges. For example, how to standardize it and ethical concerns for the clinical use of those techniques. Then there are chances of off target effects. Then delivery issues, so the struggle with the efficient delivery to the brain tissues. So in this particular in the screenshot which I have taken, so this, they are talking about the Alzheimer's disease, that's why they have given the brain tissues example here. So because the delivery is also very difficult to, means it's not straightforward, because I want to deliver it to a particular tissue, not the other tissues, so that delivery is also challenging. That is the con. Then ethical concerns, so it also requires rigorous validation due to ethical and legal concerns. And complex pathology, so editing single genes may not fully halt the complex Alzheimer disease progression. So there are many diseases which are not just monogenic, they might be due to the polygenic, means they might be polygenic depending on different genes. So in that case its pathology is quite complex. The similar thing is discussed here, that what are the potential outcomes. So for example, if this is my CRISPR Cas9, this is my guide RNA, and this is the target sequence. So if there is off target editing, then the potential outcome could be off target mutation in animals, in which it can lead to the tumor cells and tumor in mouse. And if we talk about plants, then it can lead to the abnormal gene functions, which could lead to abnormal phenotypic expression. So these are the problems which are the risk concerns related to the CRISPR Cas9 or the genome editing methods. Then comes informed consent issues. So means informed consent is very important, but still it also has certain issues. So what are those issues? The embryos and the future generations cannot give consent. For example, if I want to edit my, if I want a designer baby, so in that case, from whom to take the consent? Because I am the mother, so my child is not here yet. So in that case, the embryos and future generations, they cannot come and give the consent. So that is also one of the issue in the informed consent. Second is parental consent cannot cover unknown long term risk. So for example, some parent cannot give consent. What if in the long term, even if the parent dies, but their child has to live after that also? So we parent consent is not just enough. It cannot cover for the unknown long term risk. Then it also raises the questions of autonomy and intergenerational justice, because the what is about the autonomy, because that particular so in this case the informed consent is discussed in the terms of the babies, or the people, the means, yeah, the babies or who are not yet born. So about them, that what about their autonomy, what about their own choice, and what about the intergenerational justice, because their parents are means deciding them it for them. So there is some intergenerational justice which is required here. So these are the issues which arises. So informed consent means you tell about the nature of treatment, alternate care, benefits, risk, and any other questions which are related. So they come under informed consent. So there is a mini review which talks about the assignment of responsibility for creating the persons using the germline genome editing. Okay, so this was discussed in terms of the germline genome editing, because in germline genome editing only the embryos and future progenies come into picture. Otherwise, in somatic cell genome editing, it do not get inherited. So in that case, informed consent can be taken straight away from the person on which we are going to perform those experiments. But in the case of germline genome editing, so there are some rules which exist. So this is the example which was given: the practitioner. So prior explanation of risk and burdens is given by the practitioner. Then parents decide and they give the informed consent. Then perinatal care is given by the practitioner, and if there is some damage, then parents can claim that damage. Okay, so informing the offspring of germline genome editing. So once what parents will do, they will inform their offsprings that okay, on you this experiment have been performed. They cannot hide this information from those people. Okay, then yeah, so practitioner is doing the risk assessment, and then he's practicing, he's performing the germline genome editing. Then genome edited babies will be generated. Then infants, means they will grow up into infants. Now what can happen is they can ask, they can ascend the practitioner. Then later on, when they grow up to 18 plus, they become adults. So what they can do is um, they can claim the damage based on the wrongful life against the practitioner and parents. And they can also consent and withdrawal. So they can give their consent and they can withdraw from the experiments, because there are certain experiments which might means keep on going on, means they are not just one time. So in that case, they can withdraw them from those experiments, and they can also give consent that they whether they want to continue or not. So this generally takes place, for example, if we perform the genome editing on a kid who is just 10 years old, who is unaware that whether he should go for it or not. So in that case, we cannot take the consent from that child. So in that case, even if the child is means infant, so in that case we take the permission of the parents. So when they grow up, then they can decide whether to withdraw from that particular experiment or whether to give the consent. So this is how it talks about that, how it is regulated, how the problem of this particular informed consent issues are dealt, because there is no other way to do it. So follow up of growing and aging persons born by a GG and their progeny of for decades, lifetime or generations in cooperation with parents or the grandparents if parents are not there then. Okay, so this is how the experiment germline genome editing experiments are performed, that till the time the babies are not born, the informed consent is given by their parents or their grandparents, whoever exists. Okay, second point was justice and equity. So possible creation of genetic haves and have nots. So wealthy may access the enhancement technologies first. Bioethics demands the fair distribution of medical benefits. So that's how the justice and equity point comes into picture. So now what is the difference between the justice, equity and equality? These are the three terms and they have different meanings. So when we say equality, then we means that everyone benefit from the same support. So in this case, they all have the wooden planks which are kept here. So they all are getting the same plank. Now it depends on them how they can get benefited. So that means they all are given the same support. So this is the equal treatment. Second is equity. Equity means that everyone gets the support they need. So in this case, he do not need any support. So he's given one block, and in this case, that particular person needs two blocks. So in this case, the support is given in such a way that what is required. So this is the concept of affirmative action, thus producing the equity. Then what is justice? So justice means all of them. For example, it is an example in which they are seeing the game. So the cause of the inequity was addressed. So why they were not able to see it? So that cause is being addressed. So now they have replaced this wall which was previously wooden wall with this particular net wall so that everyone can see without support. So they have removed the reason behind the inequity. So that is the difference between equity, justice and equality. And there is a perspective, this is just an article which was published which talks about the advancing genomics to improve the health equity, that how we can improve this health equity. So by the equitable participation, measuring the health equity, policy development, partnerships and diversity of genomic workforces. Okay, so this particular figure talks about measuring the health equity, policy development, so all these points which I mentioned. So in this case, all these points are need to be kept in mind when we talk about the bioethics. So there should be equity whenever some genomic experiments are performed. So everyone should get benefit. Okay, long-term monitoring. So because we discussed the disadvantage that you might not know the long-term effects, so there is a requirement of long-term monitoring. So germline edits require the observation over generations. Participants cannot be forced into the lifelong follow-up. It raises the data privacy, consent and responsibility issues. Okay, so these are some of the excerpts which I obtained from the articles. So this was taken from the PEW Research Center. It talks about the public, especially concerned about the germline editing, that how much public is. So this is about the US. So on the US adults, the survey was performed. So it talks about the percent of US adults who say that the gene editing giving healthy babies a much reduced risk of serious disease would be more acceptable, less acceptable, or make no difference. So in this, they have performed a survey that how many people believe that after performing the germline editing, the children, babies were healthy. They were not having any severe disease. So it should be acceptable, or this particular genome editing should not be acceptable. So if effects were limited to that person and could not be passed on, so in that case, it was 34% people were mentioned that it is more acceptable. 23 believed it is less acceptable, and 40 believed that there is no difference. Then the another was that if it changed the genetic makeup of the whole population. So what if that particular change gene editing, if it is making the whole population changed? So in that case, 17% says that it is more acceptable, 49 says it is less acceptable, and 31 says there is no difference whether you do it or not. So with this, they mentioned that means if germline editing is performed, then it should not influence the entire population, because as it is irreversible. So based on it, many people think that it is less acceptable in the case where it is changing the population, and it is more acceptable if it is just limited to that person and it is not getting passed on. Okay, similar thing is discussed here, that whenever we are designing some rules or everything, so we require critical thinking, which talks about the anthropology, in which we study how the humans are distributed. We discuss about the sociology, biology. These all things are need to be kept in mind. Their knowledge and vision is very important to decide that what should be the future. So this comes under the long term monitoring. These all things are monitored for better future. Okay, similar thing is about the research ethics and biosafety. So embryo research restricted under the national and international laws. US Dickey Wicker Amendment 1996. So this US Dickey Wicker, they are the two people. One was this and other is this. So one is US. Sorry, I hope but I don't think I've written the okay, yeah, I've written their full names. So one is Jay Dickey and other one is, so he is Jay Dickey and he is Roger Wicker, who is the Mississippi senator. So these two people, they came up with the amendment, is a change means in the law. So US Dickey Wicker amendment, the name was given because these two people were involved. So they mentioned, so they have made the amendment in the US law to that prohibits the federal funding for the research that creates or destroys the human embryos. So they mentioned that government will not fund if the scientists are performing experiments related to destroying the embryos or creating the new embryos. So oversight by the institutional review boards and the biosafety committees. Okay, so same thing is mentioned here also, that they mention that government will not fund. Even if you want to perform that experiment, you are free to perform, but but not from the federal funding. Okay, so what is the meaning of regulation? So regulation means the official rules which ensures the safety and compliance in biotechnology. It oversee research, manufacturing, import, export. So in all these things, there are some rules which need to be regulated. It protects the environment, human health, and ethical boundaries are also established with the help of those regulations. Okay, so same thing is discussed that if in terms of engineering the microorganisms, so if you are performing the microorganism engineering, so in that case we also need to keep these things in mind, that whether it should be having the effect on the ecosystem, what impact it would have on the biodiversity and environmental containment. So all these things are kept in mind. So genetically modified organisms, if they are used in the antibiotic production, then also these things are kept in mind. There is a bioethics and regulatory considerations before doing any such experiments. So the role of regulatory agencies is to ensure the safety, ethical considerations, compliance and enforcement, public engagement, and international collaborations. So these are five main roles of the regulatory agencies which design the regulations so that the environment is protected, biodiversity is protected, the rights of the people is protected, and experiments are performed in the ethical boundaries. So it comes under the regulation. Okay, now we'll talk about the history, that how it started. So in 1973, the recombinant DNA raised the safety alarms. So at that point of time, people realized that there are some issues which can happen with the recombinant DNA technology. In 1974, NIH set up the Recombinant DNA Advisory Committee. So in this committee, different scientists who were well, means known to this particular field, they were there in this committee and they discussed that what are the ill effects of the recombinant DNA technology, what are the pros, what are the cons. So on the basis of that, later on in 1975, this Esilomer conference was conducted in which they created the biosafety guidelines, which was like the model for the global regulation. So globally, different rules were established, different guidelines were written, that what are the guidelines need to be followed while working with the recombinant DNA technology. Then comes growth of regulatory frameworks. So in 1980s and 1990s, international discussions on risk and ethics takes place. OECD 1986, in which they introduced the recombinant DNA safety considerations. And similarly, US, Europe all performed the product based evaluations and assessment. Then US regulation timeline. So in 1986, they coordinated the framework for biotechnology. In 1996, Dickey and Wicker Amendment, which restricts the embryo research with the federal funding. So that is also important point here. So in 2017, NAS guidelines allow the gene editing under strict conditions, and in 2020, FDA updates gene therapy safety standards. Okay, so this is all about the historical things. These are just the facts. Then US agricultural policy came up in 2018, which declares some CRISPR crops as equivalent for, means they mentioned that these CRISPR crops are as same as the conventional breeding. So conventional breeding and the genome editing, there is a difference between both of them. So conventional breeding is allowed, but gene editing is not allowed in some countries. So example is known browning mushroom and waxy corn. So no regulation if no foreign DNA is inserted. Now we'll look into that difference. So this is genetic engineering and this is gene editing by CRISPR. So in genetic engineering, what we do, we have this plasmid. Sorry, we have the plasmid from the Agrobacterium tumefaciens, and what we do is we modify the plant DNA, and this Agrobacterium tumefaciens, it has the desired gene. Whenever the plasmid will be put into the host cell, so what it will do, it will that particular desired DNA will get integrated into the host DNA. So in this case, the host is the target plant. So plant will have the modified DNA coming from the Agrobacterium tumefaciens plasmid. So in that way, the transgenic crop will be having the new trait. So in this case, it is a genetically modified organism, that is GMO. So it need regulations, and if I am supplying it, so it should have the label which says that it is GMO. Whereas if I am performing gene editing by CRISPR, so in this case I am performing double strand break in the plant genome, and after the DNA repair, the NHEJ or homology dependent repair, then there is a donor DNA which is having the homology to that particular genic section, so that get inserted, or with the NHEJ it can also get deleted. So there is just one base pair change, and this is the crop with the modified or the new trait based on what kind of repair mechanism took place. So in this case, whether it is a GMO or non-GMO, so that is a question, whether it needs regulations, whether we should label it as genetically modified, because we are not providing any external DNA, we are just editing what was already existing. And in the case of here, where the donor DNA is provided, so that donor DNA is also the corrected copy of that same gene only, that this gene is not foreign gene. So in this case, it's very difficult to decide whether it is a GMO or non-GMO. So there is a long-term assessment which is required while designing the regulations, that on which species the regulations, these rules will be applied and where these rules will not be applied. So in the case of US, they mentioned that these CRISPR crops, they are like conventional breeding. There is no requirement of regulations, and there is no need to mention them as genetically modified. Then regulatory development in India. So in 1989, different rules for the genetically modified organism came up, in which biosafety and approval processes lie, that whenever you are doing such experiments, so it should have the biosafety measures and approvals to be followed. In 2000, ICMR guidelines which ban the germline therapy. So in 2000, germline therapy was banned by the ICMR guidelines. In 2003, Cartagena Protocol ratified, and in 2019, gene therapy guidelines for ethical clinical trials came up. Then gene editing research in India. So focus area is hemophilia, sickle cell anemia, and beta thalassemia. So these are the disorders on which the research is carried on. Leading institutions are IGIB, NCCS, NCBS, INSTEM, and there might be some other as well. Emphasis on therapeutic and not enhancement applications. So in India, mostly whenever gene editing is performed, so it is just for the treatment, for the therapeutic purposes, and not for enhancing some features or doing other studies. And other reason is also that India don't want to waste its money on the useless experiments. So that is also one of the reason that we focus on those things which are the high priority things. So there is another article which I obtained from the Forbes. So in that, this report is mentioned, which talks about the GMO research, review and regulation, that how does a GMO get to the market, what are the different things which are considered. So there is a regulatory science which exists, in which it is checked that whether the crop is safe to grow, whether it is safe for environment and beneficial for the insect. Because if I am editing a crop to increase its yield, so what if an insect who is dependent on that crop, what if that insect eats the crop and dies? So in that way, I am creating an imbalance in the environment. Although the such crops exist, such as BT cotton, to avoid the um, ball worms which generally eat up the cotton. So in that case, we make the BT cotton, which is which is resistant to those insects. So in that case, somehow it is beneficial for us, but it is harming those insects who were dependent on it. So means all these things are assessed, that whether it is safe for that balance and it is safe to eat. So this come under regulatory science, and then regulatory review. So in this, the different countries, so 90 government bodies, even more than that, they review and approve the GMOs. And in many countries, multiple agencies are also there, which regulates that whether that particular GMO should go into the market or not. So GMO have been grown and imported by the 70 countries since 1996. So there are certain genetically modified crops which exist, or genetically modified organisms which exist. Okay, if we talk about China and Japan, so in China 2003 they ban on the edited embryo implantations. In 2018 there was a scientist named He Jiankey. So there is a case in which he did certain experiments which we will also discuss in later slides. So he was present for it, and he was also imposed on fine was also imposed on him for performing those experiments. And in 2020 civil code strengthens the gene protection. So civil codes were laid which were further helpful for the protection of the genes. In Japan, 2015 to 2020, gradual acceptance for the medical and agricultural editing took place, and chimera studies permitted for the organ research. So this is about the China and Japan, how they work to that. So other regions like Europe, they strict the GMO laws, including the CRISPR products. So in the previous slide, as we have seen, that US says that the CRISPR products are like the conventional breeding methods only, so no regulation is required. Whereas Europe says that the CRISPR products are also genetically modified, so there should be strict laws on those products as well. Africa Biosafety Act in Nigeria, Kenya, Senegal support the food security. If we talk about Australia and Canada, so it is license based oversight. Canada regulates by trait and not technique. So they says that whether the trait is beneficial or not. So at that, if that is beneficial, then it allows the means generation of those genetically modified organisms. Otherwise, it will restrict it. It is not dependent on what technique is getting used. Okay, so CRISPR embryos and the law. So this talks about different countries and how they look into the CRISPR embryos, whether they ban or they are banning it with some guidelines, or it is ambiguous, or it is restrictive, and in some country it is not surveyed. So as far as India is concerned, it is, I guess, banned with the guidelines, because we have seen in the previous slide that India in 2003 they banned this thing. Okay, so key regulatory challenges. So there are different definitions of GMOs and lack of harmonized international standards, balancing innovation versus public safety, managing public trust and transparencies. So these are some of the challenges which are associated with the regulatory challenges, because before imposing the challenge, means when I'm writing the rules, that this particular rule is applied to this particular organism or the species. So in that case, what is the definition of the subject on which the rule is applied? So that is important in this case. That is, the example is the GMO. So when we say that this particular crop is GMO, so due to the differing definitions, because each country has its own definition, so there is lack of international, lack of means one single definition of GMO. So that's why these are some of the challenges which are associated. And this talks about the principle for the responsible governance of gene editing, such as transparency, government regulations, voluntary best practices that complement the regulatory oversight, risk avoidance, robust, inclusive access to technology and resources. Okay, so this talks about the opportunities and the challenges which are associated. So opportunities which are associated with these techniques is the targeted medicines, biofuels, disease resistant crops, new industrial products. So all these are the opportunities which the genome editing provides us. But the challenges of the safety, ethical concerns and regulatory challenges. Okay, so now ethical foundations in genome editing. So previously also we talked about different 7 points. So here also there are main 4 points. First one is autonomy, second is beneficence, third is non-maleficence, and fourth is justice. And then there is also responsibility. So these points keep on changing, but just keep in mind that what the meaning of each point is, and each point is the foundation. These are the ethical foundations which are important whenever we are writing the regulations or rules, and these things are kept in mind. So autonomy means that the person has the ability to self-govern. So in this examples are also given for so that you can understand it better. So example is, for example, the human fetal and embryological collections. This would be providing the next of the kin with all the information to facilitate an informed decision making process. So this is the example that uh, autonomy means that the, for example, if parents are giving the consent to perform the experiment, but later on the all the information should be given to those kin, means those kids which further on grow up, so that they can take the informed decision of their own. Beneficence means acting in the best interest of the person. That means example is that, for example, for human fetal and embryological collections, this would be acting in the best interest of next of kin. So in this case, the example is given with respect to the embryological thing, but beneficence simply means that uh, the person on which the experiment need to be performed, he should be benefited out of it. So that is beneficence. Non-maleficence means obligation to do no harm. So for example, similarly for human fetal and embryological collections, this would be ensuring that all the processes, they do not create the harm to next of kins or fetal and embryological remains. Okay, so that is non-maleficence, that it should not harm these fetals or embryological remains. And justice means treating all the persons with fairness, equality, and impartiality compatible with the legal statutes. Uh, so, for example, for the human fetal and embryological collections, this would be treating NOK with fairness, equality, and impartiality. So, justice means treating it equally. Then there are major ethical concerns such as safety of target mutations, mosaicism, consent, such as embryos cannot give consent. So, this is also one concern. Equity, that means risk of genetic class divide, in which we have seen that there are some people who can afford it and other people who cannot afford it. So in that way there is the equity is not established, so that is one concern of it. Slippery slope, that means therapy leads to enhancement and leads to eugenics. Eugenic means better genics. Okay, so it means therapy is one thing and enhancement is other thing. Although therapy also leads to enhancement, but if I am just enhancing a trait versus I am using that mutation for the therapy, so there is a difference between it. So many times what can happen is that on the name of therapy, some people can enhance their traits which might not be good thing. So that is a slippery slope here, that how to decide, what should be deciding criteria. So these are the major ethical concerns, and these are some of the other concerns as well, such as conflict of interest, ethics of privatization, confidentiality of patients, equity of resource distribution, and patients' rights. Okay, now we'll talk about what is therapy versus enhancement. So, therapy means when you correct the defect to restore the health. Enhancement means you improve the normal traits, just in just like intelligence, strength. So, there is an ethical question that where to draw the line, because in the previous way where we have discussed the term that there is a slippery slope, that is the ethical concern. So this is the question that where to draw that line. So most regulations allows the therapy only. Okay, so these are the types examples of human enhancements. So phenotypic, somatic, and germline. So phenotypic means you improve the body without changing the genes. For example, bionic limb, special glasses, and exoskeletons. For example, if some person is not having one leg, so in that case the bionic limbs are given to him. So that is the phenotypic enhancement which is widely accepted. Or such as special glasses, if somebody is not able to see, so the glasses which help that person to see, so that are like the without changing the genes, externally physically some enhancement is done, so which is widely accepted. Then somatic means which are non-inheritable, so it changes the body cells but they are not inheritable to the future progenies. So for example, gene therapy for diseases which are the somatic cell modifications or targeted cell treatments, so this is also mostly acceptable. Then comes germline enhancements, which are heritable. So it alters the embryo genes, affect the future generations. For example, embryo gene editing, heritable modifications, or designer babies. So they are major ethical concerns. Okay, similar example is given that therapy. So disease causing mutation is there, After CRISPR-Cas9, disease-causing mutation is removed, so it leads to healthy wild type. Second is genetic protection modification. So in this case, there is a risk variant with the help of CRISPR-Cas9, healthy wild type without the risk variant. And when we say enhancement, so the healthy wild type was there, but with the help of CRISPR-Cas9, we made it a superior variant. So this is known as the enhancement. So this kind of enhancement is not allowed. It's generally prohibited. And then there are some socio-economical and the cultural issues. So many people, um, cultural means that many people who believe in God, so they also says that these genome editing techniques, what they are doing is they are like playing God. You are changing the genes, you are changing the traits. So in that way, this is people who believe in the who are the religious, they do not accept these techniques. Second thing is that gene editing may widen the rich poor health gap, because these editing techniques or these things, they are costly. So rich people will be able to access it, whereas poor people will not be able to access it. So in that way, it will also increase furthermore the gap between the rich and the poor at the level of their health. Then cultural and religious values also influence the acceptance. So most highly religious Americans say gene editing of babies would be meddling with the nature. So this is also another survey obtained from this particular article. So in this, they performed a survey on the religious families in America, and they discussed that whether the gene editing performed on baby should be acceptable or non-acceptable. So there are two categories. One is that is no different than the other ways we try to better ourselves, and second is crosses a line and is meddling with the nature. So US adults, 51 out of them says that it is not different than the other ways in which we try to improve ourselves, whereas 46% believes that if we perform gene editing, then we are trying to interfere with the nature. And among those on the religious commitment index, the people who were highly religious, so 33 of them says that it is not different and it is good for our betterment, whereas 64% says that it is interfering with the nature. The people who were medium religious, so 49 of them were means ready to accept this. 48% says that no, this is playing with the nature. And the people who are having the low religious commitments, so 70% of them says that yes, gene editing is making us better, so it is acceptable, whereas 28% of them says that um, no, it's like messing with the nature. Okay, so this is how the, because whenever some uh, techniques come up, the policies are designed. So policies are not written by just the people who are the policy makers. So these policies are also written by looking at the consensus public, that whether that public is ready to accept it or not. So even if the technique is very good, it is providing us therapy, it is means having the therapeutic benefit, means it's beneficial, but still if people are not able to accept it, then that particular technique cannot come in market, or it cannot be allowed. So means this is the survey is one such way in which the public opinion is collected. So means that's why it is important to keep in mind that whether to allow the gene editing techniques freely, or whether we should put some restrictions, and what kind of restrictions and on what things restrictions should be put. So this is means defined on the basis of public opinion. Okay, ethical issues in embryo editing are the risk and harms, informed consent, that is one issue which we discussed, that parent versus child, then justice, equity and social considerations, eugenics, that harm to person with disabilities or it is leading to enhancement, then oversight, the regulation and policies, ethical guidelines, public engagements. So these are required whenever the embryo editing is done. Then what are the benefits? Whether it leads to the disease or it provides immunity to a disease, risk or harms. So it is having the risk of harm to embryos, risk of harm to the family, or risk of harm to the society. So these all things need to be kept in mind. Okay, so these are the issues which are related to the embryo editing. These things are need to be dealt with. Then comes environmental and agricultural ethics. So CRISPR crops challenge the traditional GMO definitions, because as we have seen that uh, CRISPR crops, they are not bringing the foreign genetic material, because with the help of those recombinant DNA technologies, what we do, we use the plasmid in which we have the gene of interest. So that gene of interest is the foreign gene that is not belonging to the same host organism. So in that case, that is called as the GMO, genetically modified. But in the case of CRISPR crops, there is a challenge because we are not bringing the foreign genetic material into the host. So there is a challenge to establish the definition for that. Then potential risk is of the gene drift, ecological imbalance, and cross species transfer can also take place. Then the regulatory questions are that should this transgene free plants be regulated as GMOs? Because in this case, CRISPR crops, there is no transgene. So it is a transgene free plant. So whether there should be same regulations as that of GMO or not, this is one another question. So ethical questions related to any genetically modified crop or any crop is environmental safety, monopolies and patent controls, and consumer transparency and labeling. So with respect to environmental safety, for example, if some crop comes in market after the gene editing, so it should be safe, it should not impact the environment in the wrong way. Then comes monopolies and patent control. So there are some uh, communities or some group of scientists or some organizations, for example, they bring some product to the market, but what if they get monopoly of it? So they will not allow the others to produce the same thing, and in that way they will, if what if they have their patent for that particular crop so that nobody else could use it without their permission. So those are the ethical questions. And then consumer transparency and labeling, for example, somebody is going to market to buy those crops, so they should be informed that it is GMO or it is how it is produced. So that is also another ethical question. And this is the another screenshot which I obtained from some article in which they have discussed about the when to regulate something as GMO. So there are different rules. So extent to genetic changes, that is measured to decide that when to regulate it. So this is the green one, shows that it is regulated as conventional, that means non-GMO. The conventional means normal crops. So if there is natural mutations or if there is mutagenesis, if mutation is performed in the same gene, so in that case we'll consider it as non-GMO, so there will be no regulations. Then comes changes possible with the genome editing, such as no template or small changes and deletions, such as in the CRISPR-Cas9 when we are repairing it with the NHEJ method. Then short template including the ODMs, and long repair template which is cisgenic. So it could be created by the conventional breeding and there is no foreign DNA which is used here. Now there is a middle which says the transgenic repair template. So this means that when we are performing homology dependent repair, then the template which I am providing, the external donor DNA, that is a transgenic, that is from the another gene or some foreign particle. So in that case it includes the transgenic DNA sequence. So it means this is the part where there is a challenge, or the where there is exist a challenge that whether to consider it as GMO or not. So some countries consider it to be GMO, some countries consider it to be conventional. And then comes the recombinant DNA insertions. So they are regulated as GMOs and include the transgenic sequences. So this talks about extent of genetic change. So towards the right side, that means there is more genetic change which has occurred. On towards the left side, the less genetic change. And then risk and balance which need to be maintained. So the risk is the food safety risk of meat from the healthy animals, food safety risk of milk from the healthy animal, risk to animal health, increased risk to environment from the farm animals. So in context of the animals, genetically modified organisms, so these are the risk which are there. Means these are the questions which arises, that whether whenever we are performing the genome editing, that whether the because the animal many countries consume the meat of animals, including our country as well. So in that case, if you are editing, you're performing editing on that organism, so whether their meat is safe to consume or not, or whether the milk is safe to consume or not, or whether that genome editing is harming the animal or not. So there are some these questions, still the risk which exists, that which decides that whether to approve that gene editing or not to approve. And potential benefits include disease prevention, improved animal welfare, reduced zoonoses, and reduces antibiotic use, reduces impact on the environment, adaptation to climate change, and fewer resources to produce milk and meat. So these are potential benefits of approving. Okay, then there are some other animal welfare and chimera research. So creation of chimeric animals raises the moral questions. So chimeric means when you are having the different genes and different genes combined, so that makes a chimera combination of things. So that is known as the creation of chimeric animals. So the moral questions which arises is human cell integration. So it leads to identity issues, welfare concerns due to off target effects, and then debate continues over the limits of animal experimentations, because if you cannot directly perform experiment on human, then you exploit the model organisms. So there should be some limit also, that to what limit you can perform those experiments. So still debates are going on, the people who are associated with the animal welfare, so they said that there should be some limit, that you should not be free to exploit the animals for the research. So how chimera research could create the organs, because this research is good also, this is not just bad. So chimera means that I am taking some cells from the animal and then I'm using some cells from the human, I'm combining them, so that is like mixture of the genes. So injecting human stem cells into the animal embryos could lead to an unlimited supply of human organs, because we know that stem cells are the cells which are totipotent in nature, which can give rise to, which can get differentiated into any of the cells in the body. So what we can do is, if that stem cells are um, injected into the animal embryos, then it can lead to the production of any organ in the body, so which can help us to generate any human organ. So there are now long waiting list of organs, and many people who need them die waiting for that to take place. So human stem cells, they have the potential to become the cell of the any organ. Then in pig embryos are taken, human stem cells are injected into the pig embryos that lack the genes to create a specific pig organ. Pig act as the human incubator. Human organs grow as the animal matures. Organ transplantation, organ is transplanted from pig to the human. So this is how the chimeric research create the organs. But at the same time, we are exploiting pig. Okay, so I don't think this is that much common practice. It is very rare, and still different experiments or different things are going on, because this is not very common practice. But this is how the chimeric research can lead to the generation of organs. So after this organ get matured, it can be taken from that organism and put into the human. But animal welfare committee should not accept it, because this is doing very wrong thing to animals. Okay, similar thing, this, if you're interested, you can go through this screenshot in which it talks about the microbiome ethics. So in this they talk about just the microbiome, which is the microorganism inside the human body. So there are ethics towards that as well. Whenever we are modifying them, then different things we should keep in our mind. So this is about it, I'll not go into the detail. This talks about the same thing, that whenever you make any changes at the level of genome, so it should not harm, and different seven laws or different seven principles they should be followed during this particular genome editing experiments. And then equity and access, so who decides what traits can be edited, will editing be affordable and accessible globally. Ethical goal is to avoid creating a genetic elite. So as we discussed previously, that the people, rich people, so it should, means the regulations are put in such a way that the genome editing should not be exploited to generate, to perform the enhancements, rather it should be used for the therapeutic purposes. So this all questions come under the bioethics, that whenever we are designing the regulations or rules, then these questions are asked. Okay, so equity in the global access to the genome therapy, research and development. So these are different questions: global health justice, fairness, inclusiveness, best interest of the child, engagement. Then regulatory system should be transparent, openness, scientific evidence based decision making. So whenever the regulatory system is deciding or writing some rules, so there should be some scientific evidence to it. Social and legal accountability, because accountability is very important. For example, some people have allowed for the experiment to be performed, and later on they get adverse effects. So in that time, this regulatory system should be hold accountable for this. They should give means compensation to those people, so they are accountable for it, and maximizing benefits and minimizing the harms. Then manufacturing capacity, sustainability, social and legal accountability, responsible stewardship of science and delivery, social justice, resilience and efficiency, inclusiveness, engagement. So these all come under the equity practices, and these are the different components of it, that how the equity is managed in the genome therapy and how its global access is managed. Okay, so the case which we discussed previously of the He Jiankui in 2018, so he was imprisoned. So he was the Chinese biophysicist who gained the means who attended the international conference in 2018, in which he announced that he has generated world's first genetically edited babies using the CRISPR-Cas9 technology. So his goal was to edit the CCR5 gene, because we have also discussed that to treat the HIV, if we edit this gene, because this gene is responsible for the receptor creation on the cell surface, and that receptor is responsible to bind to the HIV. So if we edit this gene, then receptor will not be formed and HIV will not be able to bind to the cell surface and insert its genetic material. So in that way, it that cell is resistant towards the HIV infection. So his goal was to edit that gene, and this case sparked the widespread ethical and scientific debates. So Chinese scientists edited the twin embryo to resist the HIV, but global condemnation for violating the ethical and legal norms, because in this case he is editing the germline cells, and outcome was that he was imprisoned for 3 years, and about 3 million fine was imposed. So 3 yuan is the Chinese currency, so that million fine was imposed on him, and this sparked the global moratorium on the reproductive gene editing. Okay, so this was the news which I obtained, so in which it was mentioned that although the experiment which he performed is related to the therapeutic purposes only, but still the rules or regulations he disobeyed them, so that's why he was put in prison and he was also fined. So he thought that he is the first one to do it, but at the cost of this imprisonment and fine. Okay, so now the conclusions and what are the future directions. So genome editing, although it is very powerful, but it is very ethically sensitive tool. It requires transparent and globally coordinated regulation, and ethical use must prioritize safety, equity, and human dignity. And for the future, so international governance for gene editing standards are required. Okay, so these plots also discuss the same. So knowledge and understanding of the cells and gene therapies, acceptance of cell and gene therapies, chemotherapy concerns, religious beliefs, potential benefits, source of stem cells and vector, understanding the risk and benefits of the therapy, and information needs and sources. So these all blocks, or these all circles, they are linked to one another, and these are required, means whenever we are performing the genome editing, then these all things are need to be kept in mind, and different regulations and rules, they are designed on the basis of all these bioethical principles, that it should be fair, it should provide justice, the organization which is doing it, they should be transparent enough, it should be beneficial, non-maleficence, that it should not harm anyone. So all these points should be kept in mind. Okay, so with this we'll start the MCQs. So okay, we have time. Also, I hope whatever we have discussed about the ethics and rules, that is clear, because it was more like uh, the facts and things. It was not much to means about the techniques or treatments. It was about what is globally going on, and what are the ethical practices, and why they are there, why regulations are required, what points should be kept in mind. Okay, so let's start with the question. So first question is, who coined the term bioethics? Option A, Van Rensselaer Potter. Option B, Fritz Zahar. Option C, Maharishi Chark. And option D is S.K. Pandya. Yeah, so that's right. So Fritz Zahar was the one, and this was his book, From the Origin of Bioethics. Okay, next question is, what was a key outcome of the Nuremberg trials regarding the medical ethics? Option A, introduction of CRISPR technology. Option B, the development of the Nuremberg Code for the human experimentation. Option C, the approval of gene therapy for cancer. And option D is the formation of Esilomer Conference. Yeah, so this is quite straightforward. So Nuremberg trials, they led to the development of Nuremberg Code for the human experimentation, and this is the news article. So it was these trials were conducted from 1945 to 1947. Till 3 years the trials were conducted, and these were the military tribunals which were held after the World War II to prosecute the Nazi war criminals, which included doctors also who were conducting the inhuman medical experiments on the prisoners without their consent. And later on in 1947, the Nuremberg Code was developed, which set the ethical principles for the human experimentations. So these means the main points of this particular Nuremberg codes were voluntary consent, avoidance of unnecessary suffering, right to withdraw from the experiment at any point of time, and scientific necessity and benefit must outweigh the risk which are associated. So yeah, so I mentioned this thing because it is very important and it might come in your exam. Next question is, which of the following is a concern regarding the international regulation of genome editing? Option A, uniform standards. Option B, lack of expertise. Option C, cultural acceptance. And option D, differing regulations among countries. Yeah, so you're right, because as we discussed that in the case of the rules and regulation related to genome editing, it becomes very difficult because different countries have different definitions of things. So there are different regulations. That is the major concern, and this inconsistency can lead to the issues such as science tourism, in which the researchers conducting the experiments in the countries with the looser regulations. So, for example, there are some countries like Europe which completely ban the um, things which are like working with the embryos or working with other cells for the genome editing. So in that case, what scientists will do, they will go to other country where there is loose regulations, so it will promote the scientific tourism. That's why this international regulation is a concerning thing. Then ethical conflicts between the nations with the different cultural or moral views, so it can also lead to that conflicts between different nations. And difficulty establishing the global oversight for emerging technologies like CRISPR. So CRISPR technology, so it is very difficult that uniform universal laws could be established due to this particular thing, and also establishing the global oversight. So difficulty is there due to this particular reason. Okay, so next question is, what are the potential risks associated with off-target effects in genome editing? Option A, increased therapeutic efficacy. Option B, unintended genetic changes outside the target region. Option C, permanent removal of all the diseases. And option D, acceleration of germline mutations. Yeah, so you're right, because off target simply means that other than the target. So unintended genetic changes outside the target region. Okay, I have added some other points also. So these unintended genetic changes, they can lead to disruption of important genes causing the harmful mutations. Second is activation of oncogenes. So oncogenes are the genes which increase the cancer, which leads to the cancer onset. So it might activate those oncogenes. Then unexpected physiological effects that may not appear until the later in life. So that can also one of the reason. And ethical and safety concerns. So especially in the germline editing, there is the ethical and safety concern because this germ cells get inherited. So that is also one such reason. So next question is, what bioethical concern arises when genome editing is used for the known therapeutic purposes? Option A, enhancement for athletic performance. Option B, medical therapy for genetic disorders. Option C, environmental protection. Option D, eradication of infectious disease. Yeah, so that's right. Option A is the one, because other options are kind of good only, means they are more linked to the therapeutic purposes of the genome editing technique. So enhancement for the athletic performance means the person is having the normal gene and you are enhancing it. So that is one of the bioethical concern which arises. And other purposes, other things where the human enhancement takes place. So that means that examples include editing genes to improve the intelligence, appearance, and athletic abilities, creating so called designer babies, and widening the social inequality as only the wealthy people will be able to afford that enhancement, and undermining the concept of human diversity and natural variations. So because if everyone will go on designing their babies and modifying the traits, then there will be no variations. Means if there is no regulation, then everybody will do it, and then the variation will decrease and diversity will also decrease. So these uses cross the line from the medical treatment to the genetic enhancement, and that's why different debates are going on this. And means, yeah, it's the rules says that the enhancement is not allowed, but the uh, for the therapeutic purpose it is allowed, but still there is that slipping slope issue which exists, because it's very difficult to decide in certain cases that whether it is having the therapeutic purpose or whether it is leading to some enhancement. Okay, so next question is, why is there a concern about the privacy in genomic research? So option A is high cost of data storage, option B potential misuse of the personal genetic information, option C difficulty in obtaining the DNA samples, and option D is lack of scientific interest. Okay, so I think you're saying something and I don't have earphones on. Okay, so um, yeah, so the right answer is option B, the potential misuse of personal genetic information. And in this slide, I've also kept an example of the untold story of how the Golden State Killer was found, and it was a covert operation and private DNA. So, although the misuse of personal information is means it's concern about the privacy in this case because people can use it. So in this story, this particular misuse has helped the people to find the Golden State Killer. So privacy is a concern in genomic research because DNA contain highly personal data, because it can reveal the person's identity, health, risk, and family relationships. So it can be misused by the insurance companies or employers to discriminate them, and it can also lead to loss of anonymity. So example is of this particular person. So in 2018, the law enforcement they used the data from the public genealogy website to identify the Golden State Killer, showing that how the genetic data shared for the research or ancestry can be traced back to the individuals even without their direct consent. So uh, this was one example in which, although this is not allowed, but this is just to mention that this particular personal information, how critical it is that this person was arrested due to this by recovering his data from some public site where the genetic data was available without the consent. Okay, so next question is, which international agreement aims to ensure the safe handling and transport of organisms modified by the biotechnology? Option A, the Cartagena Protocol, option B, the Nuremberg Code, option C, the Helsinki Declaration, and option D is the Esilomer Agreement. Okay, so in this case, because it talks about the safe handling and the transport of organisms, so it is related to the Cartagena Protocol on Biosafety, which was adopted in 2000. So it is an international agreement under the Convention on Biological Diversity (CBD), and it aims to ensure the safe handling, transport, and use of living modified organisms which result from the modern biotechnology, particularly those that may affect the biodiversity or human health. So the Cartagena is associated with the safe handling and transport of organisms. And Nuremberg Code we have already discussed that it's related to the principles for the human experimentation, and it was laid after the Nuremberg trials. Now the Helsinki Declaration, so it gives the guidelines for the medical research involving the human subjects. So it gives those guidelines that whenever your subject is human. So it was laid in 1964. In Esilomer Agreement, which was in 1975, it talks about the safe recombinant DNA research. So it was not the international treaty first of all, and second thing, it relies on the how the safe recombinant DNA research can be carried out. So the safe handling and transport of organisms by which are modified by the biotechnology is the agreement which talks about it is the Cartagena Protocol on Biosafety. Okay, so next question is, what is the primary focus of bioethics? Option A, economic issues in medicine. Option B, ethical, social, and legal issues in biomedicine. Option C, technological advancements in genetics. And option D is political aspects of healthcare. Yeah, so this is quite straightforward. Ethical, social, and legal issues in biomedicine. Ninth question is, which country declared that the genome editing is equivalent to conventional breeding in some instances requiring no regulatory oversight? Option A, India. Option B, US. Option C, Japan. And option D is China. So which of the countries thinks that this uh, genome editing by CRISPR or any other technique, it's same as the conventional breeding only, and we should not, I mean, no regulatory oversight is required? Yeah, so option B is right, which is the United States. Okay, because United States has mentioned, whereas Japan, China, and India, they're not the ones. They There are some rules which exist. They do not treat it as conventional breeding, although the uh, regulations are not as same as GMOs, but still some laws are followed. Okay, so next question is, which regulatory body in the United States was created to address the challenges of recombinant DNA research? Option A, FDA. Option B, NIH Recombinant DNA Advisory Committee. Option C, USDA. And option D is NIH, which is National Institute of Health. Okay, so you might not be aware. So the right answer is NIH, which is the NIH Recombinant DNA Advisory Committee. So this is a specific committee which was designed in NIH only, which deals with the challenges of recombinant DNA research. Okay, so next question is, which document provides ethical guidelines for medical research involving the human subjects? Option A, Nuremberg Code. Option B, Belmont Report. Option C, Declaration of Helsinki. And option D is UNESCO Bioethics Declaration. Because it's a document. Okay, let's see. So right answer is Declaration of Helsinki. So first of all, because it was quite confusing, you might think that why not Nuremberg Code? So Nuremberg Code was the first set of ethical principles for human experimentation, emphasizing the voluntary consent. So this was the set of ethical principles. Then Belmont Report, so US document which outlined the ethical principles, in which it says respect for persons, beneficence and justice in research. Whereas Declaration of Helsinki, it provides the guidelines, ethical guidelines for medical research involving the human subjects, and it was issued by the World Medical Association. So guidelines and principles, there is a difference between both of them. So that guidelines are means given in the Declaration of Helsinki, and UNESCO Bioethics Declaration, so it promotes the global principles on bioethics and human rights in the science and medicine. So the right answer is option C, which is Declaration of Helsinki. Okay, next question is, what is the primary ethical concern about the germline genome editing? Option A, it only affects the somatic cells. Option B, it can alter the traits in future generations. Option C, it improves the environmental safety. And option D, it guarantees the therapeutic success. Yeah, so this is also straightforward. So next question is, which principle of the Belmont report emphasizes treating the individuals as autonomous agents? Option A, justice. Option B, beneficence. Option C, respect for persons. And option D is non maleficence. So in this you need to tell the one which emphasizes on treating as autonomous agents. Okay, so in this case the principle which deals with it is the respect for persons. So in Belmont report, the basic ethical principles are respect for persons, which includes individual autonomy and protection of individuals with reduced autonomy, In beneficence, it involves the maximizing benefits and minimizing harms, and justice includes the equitable distribution of research cost and benefits. So, beneficence and non maleficence comes under the same point as well. Beneficence means the benefits, and non maleficence means minimizing the harms. So, in this case, respect for persons includes the autonomy concept here. So, next question is, which event promoted the formation of Modern Research Ethics Committee? Option A, Human Genome Project. Option B, Tuskegee Syphilis Study. Option C, CRISPR discovery. And option D, Dolly the sheep cloning. So, which event, not promoted, sorry, prompted the formation of Modern Research Ethics Committee? Yeah, so you're right. It is Tuskegee Syphilis Study. So, this is the picture of the article which was published in the news. So, the Tuskegee Syphilis Study was the US public health study which was conducted from 1932 to 1972, 40 years, by the US Public Health Service, and it involved 399 African American men with syphilis who were misled into believing that they were receiving the free medical care. But in reality, they were denied the effective treatment even after penicillin became widely available in 1940s, so that researchers could observe the natural progression of the disease. And this study was exposed in 1972, causing the public outrage and highlighting the need for protecting human participants in the research. So, this led to the establishment of institutional review boards in the US, development of strict informed consent requirements, and strengthened the oversight for all human subject research. Yeah, so means this study was exposed after 40 years. Till 40 years, these 399 people, they were not receiving any medication, just because the scientists want to study that what is the how the disease could progress. So this is how the means it led to the formation of these modern research ethics. Okay, next question is, what does informed consent ensure in the biomedical research? Option A, participants are paid for participation. Option B, participants understand and voluntarily agree to the study. Option C, data is shared without permission. And option D, researchers can proceed without oversight. Yeah, so it is straightforward. That is what the informed consent means. That they understand and then they are voluntarily getting agree to that research or study. Next question is, which of the following best defines the somatic gene therapy? Option A, modifying genes in the reproductive cells. Option B, editing genes in the body cells to treat the disease. Option C, altering the human germline. And option D is using the cloning for the genetic research. Yeah, you are right. So, somatic gene therapy means we are editing the body cells and not the germ cells or reproductive cells or cloning. Yeah, same difference I have shown here, germline gene editing versus somatic gene editing, and these are the example of cells. So in germline gene editing we have sperm cells, ovum cells, and early embryos, whereas in somatic cells these are the other body cells which could be muscle cells, blood cells, nerve cells, and this is how the modification is done. And then in the case of germline editing, the edited part can get passed on to the future generation, whereas in the case of somatic gene editing, the edited gene is not passed to the future generation. And there are some issues which are also listed, so if you're interested you can go through this. Okay, next question is, which international organization promotes universal bioethical standards through the declarations? So option A is WHO, option B is UNESCO, option C is NIH, and option D is FAO. So the right answer is option B, which is UNESCO. So if we talk about WHO, which is World Health Organization, so they focuses on the global public health policy and disease prevention, and not on the universal bioethics. If we talk about UNESCO, then they promotes the universal bioethical standards through declarations like Universal Declaration on Bioethics and Human Rights. So UNESCO is the one such organization. If we talk about NIH, so this is the US agency funding the biomedical research, and it is primarily national and not the international bioethics governance. Then FAO works on the food security, agriculture, and nutrition, and it does not set the global bioethical standards. So the right answer is UNESCO, which is the international organization and promotes the universal bioethical standards. Okay, next question is what is the major ethical issue in the human cloning? Option A, genetic diversity enhancement. Option B, loss of individuality and identity concerns. Option C, cheaper medical treatments. And option D is environmental sustainability. Yeah, so this is also straightforward, loss of individuality and identity concerns. Okay, next question is what is the purpose of institutional review boards? Option A, approve the funding for the research projects. Option B, ensure the ethical conduct in the human subject research. Option C, regulate pharmaceutical prices. And option D is certify laboratory equipment. Yeah, so the right answer is option B, that is ensure ethical conduct in the human subject research. And for the option A, which says the approved funding for research projects, so this is handled by the grant agencies and not this IRB. And to regulate the pharmaceutical prices, so this is also done by the government regulatory bodies. And certify laboratory equipment, so this equipment certification is a technical and safety issue not related to IRB. So the right answer is option B. Okay, next question is what does the term dual use research refers to in biotechnology? Option A, research beneficial for both humans and animals. Option B, research that can be used for both good and harmful purposes. Option C, studies conducted by two institutions. And option D is experiments with dual funding sources. Yeah, so you're right. So option B is right. That means the research that can be used for both good and the harmful purposes. That's why its name is dual use research. Okay, so this is the term in the biotechnology which refers to the scientific work that has potential to benefit the society also. For example, developing vaccines or treatments, but it could also be misused to cause the harms, such as creating the bioweapons. And example is engineering a virus to study its transmissibility could help prevent pandemics, but at the same time, if that knowledge is exploited, then it can lead to the intentionally creating the dangerous pathogens. Okay, then I have kept something just for your knowledge if you're interested. So there is a famous dual use biotechnology cases. One such case is of H5N1, which is the gain of function influenza, in which the scientists have modified the H5N1 strain to be transmissible between the mammals. The benefit was just to understand the pandemic potential and to develop the vaccines, but risk could be that if it is misused as a bioweapon, or what if it could get accidentally escape from the lab. So in that case, the pandemic, you will observe the pandemic. Then second is of the synthetic poliovirus reconstruction, so in which the scientists or researchers they chemically synthesize the poliovirus genome. So benefit is to study the virus biology and develop vaccines, but risk is that if this virus could get escape from the lab, or what if it is weaponized, because right now the bioweapons is very modern term which is coming up. People might use the bioweapons in which they might use the viruses, and this means they can spread that virus in some country so that the population get wiped out due to the virus infection. So in that way it can get misused. Then mousepox virus experiment 2001 Australia. So in this case also the scientists were engineering the highly lethal mousepox strain to study the immunity. Yeah, so similarly other experiments are also mentioned. And similarly the SARS virus escape in the Beijing lab 2004, in which the SARS coronavirus accidentally infected at least four lab workers at the Chinese Institute of Virology. The cause was improper biosafety practices in the lab which were handling the live viruses. So that's why the biosafety considerations are very important whenever you are performing experiments with the viruses. Then there are some guidelines which you must follow. So due to improper guideline following, this particular virus affected four lab workers. Outcome was that lab was temporarily closed and safety protocols were overhauled. Then foot and mouth disease virus in UK 2007. So in this case, virus escaped from the veterinary research facility at Pirbright, which caused the leaks from the waste handling systems. And yeah, some people do believe this, that SARS-CoV was, yeah, yeah, but it's just a conspiracy report, not published Similarly, the smallpox talk incident in UK 1978. So in that case also, Janet Parker, a lab technician, he was exposed to that smallpox in the research lab. The cause was the airborne virus transmission from an improperly sealed lab, and outcome was that particular lady, the lab technician, she died from the smallpox, and the incident was prompted worldwide reviews of lab containment practices. Yeah, so these are the some of the things which happened, due to which it was realized that how important is biosafety thing, and then later on other regulations were imposed. Yeah, but at this time also there are some biosafety rules that existed, but due to the improper following of those rules, and it's not improper but not following those rules properly, led to these incidents. Yeah, okay, then I think I have just two, three questions. Maybe this last question. So, what is the central ethical concern in the synthetic biology? Option A, lack of computing resources. Option B, creation of artificial life forms and unpredictable consequences. Option C, high cost of sequencing. And option D is limited international collaborations. Yeah, so right answer is option B. That is, creation of artificial life forms and unpredictable consequences is the major ethical concern. Yeah, so this is all about today's session. So now we can proceed with the doubts or the questions. So now you can ask the question, Tikeshwari, which you were mentioning in the beginning. Yeah, and one more thing which I want to add is that on Thursday also we'll be having the revision session. So I don't know whether they have mailed about it or not. So I'll check for it. And on Thursday also we'll have the session from 4 to 6, in which we'll discuss all the 12-week MCQs, because the in the exam the pattern is like MCQs will be coming, so that's why we'll discuss all those MCQs, so that you get to revise and summarize all those topics whichever we have covered in 12 weeks. Okay, so I think the questions will be informal only, so I'll end the recording and I'll just wait for the questions. And good luck and thank you very much.